Okay, so today we are going to talk about fatty acid catabolism, which is chapter 17 in your Leninger text. And so we have divided this chapter into two parts. So we will try to cover the first portion of it, about 30 slides. And then we will do the remaining, I think, next week. Okay. So again, I want to just bring your attention to the three stages of metabolism that we have been talking about. And you will see it's shown here on the right-hand side, stage 1, stage 2, and stage 3. Stage 1 is where you produce acetyl coenzyme A, either from amino acids, proteins, sugars, or fatty acids. And that acetyl coenzyme A is channeled through the central hub of metabolism, which is citric acid cycle, where it is broken down in steps. And in that process, you release carbon dioxides, you're reducing electron carriers, reduced electron carriers, NADH and FADH2, and those reduced electron carriers in stage 3 go on and transfer those electrons to the respiratory chains that lie in the inner mitochondrial membrane. And that process generates a proton gradient, and that proton gradients provides the energy for the synthesis of new ATP molecule. Where do the electrons end up from those respiratory chains? They end up with oxygen, molecular oxygen. So they actually reduce molecular oxygen to water. Okay. So we are going to talk about in today's class how we take a fatty acid and metabolize it to generate acetyl coenzyme A. And I show this slide to students to get them to appreciate how fatty acid metabolism is, could be crucial in certain types of function. And you see here in the center, this is American golden plover. It basically flies from Alaska all the way to Hawaii nonstop. Okay, and that's about 3,300 kilometers. Okay, so you can divide that by 1.6 and you will get the number in miles. And it flies nonstop in about, covers that distance in about 35 hours. And to cover that distance, it actually literally has to have almost 250,000 wing beats. So you can see lots of energy will be needed, right? And so there are birds like this, for example, ruby-throated hummingbird that also flies nonstop across the Gulf of Mexico. And the reason these birds are able to do that is that before flying, they actually feed on certain types of seeds. And you know that from the last class, seeds are very rich in fat, right? Lipids, triacylglycerols. And so when they feed on these seeds, they actually pack a large amount of neutral fat. And they utilize that fat to license their flight all the way nonstop. Okay, so when they're ready to fly, you'll see that 70% of their body weight is fat. Okay, so yeah, fat is a very, very important type of metabolic fuel that you can actually utilize for sustenance. Okay, so always question we ask is why fat is the preferred form of energy storage, right? And we talked about that a little bit. Just to give you a little perspective, in some types of mammalian tissues, such as, let's say, liver primarily uses fatty acid as fuel, okay, and heart, okay. 80% of the energetic needs for heart and liver tissues is coming from the oxidation of long chain fatty acids. 80%. So you can see how important it is for your energetic needs. And I also want to at the same time say that in males, mean percentage body fat is typically in the range of 23% when you are young, say between the age of 16 and 19 years, and when you are at the age of 60 and 80 years, that fat amount grows by another, let's say, 7-8%. So it will be about 31% by the time you are 60 to 80. In female, the same, between the age of 8 and 11, the body fat ranges for about 32%. And then around 60 to 80 years, it's about 42-43%. So women have higher proportions of fatty tissues. And that makes sense because women have to bear child and things like that. It's really useful form of energy. And so the main reasons for the energy storage, for preferred molecules for energy storage is fatty acid, because that the carbon in fatty acids is in the most reduced form, right? And so when it is in the most reduced form, when you are trying to do oxidative metabolism, you can expect that it will generate most energy possible. Because fatty acids don't need to be hydrated, they are fairly hydrophobic molecules, right? And that will not be the case for glycogen or sugar like glucose, right? So they can pack more efficiently, more closely in storage tissues. So those are the two reasons why fatty acids are used by many organisms to store as energy storage fuel. Okay, so with that, I also want to just tell you that most of the storage fat that we have in our adipose tissues are triglycerides. And it turns out it's not so great, but it turns out that triglycerides, which will be neutral fat, represents the major energy input in the modern American diet. Okay, so we eat lots of neutral fat. And so so that's something that that's one reason we actually one way we are consuming fat. And then you also know that once fat is stored in, let's say, adipose tissues, you can mobilize the fat using hormones like glucagon or epinephrine and adrenocorticotropic hormone, ACTH, what we call. And well, ACTH, also known as corticotropin, it's a basically peptide hormone. It's called tropic hormone. It's produced and secreted by anterior pituitary gland. Okay, and it's a very important hormone whenever there is a stress signal for the body. Okay, and it works in the neuroendocrine system. But anyway, the hormone that you are most familiar with is glucagon and epinephrine, right? And glucagon, if you remember, is a hormone that is produced by the alpha cell, right? And it plays a very active role in allowing the body to regulate the utilization and maintenance of glucose level in the blood, right? But it turns out that glucagon also plays, and epinephrine also plays a role in fat metabolism. Okay, so if your energy levels are low, let's say low blood glucose levels, glucagon is released, and that actually will allow if there is a glycogen to be mobilized so that you can normalize the glucose level in the blood, or if there is a need for carrying out glycolysis, you can actually um, 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 or if there is a need to produce energy, you can actually utilize glucagon to mobilize fat from the adipose tissues. So yeah, glucagon is a very, very important hormone. Um, it will stimulate liver to break down glycogen, right? And it will also, if you recall, will activate gluconeogenesis, where you can utilize even amino acids and convert them into glucose. And then it also is utilized in breaking down of the fat from the storage tissues such as adipose tissues. So just, we already know that, but you have to keep those things in mind. So yeah, if you look at this figure, it actually is an SEM image, scanning electron micrograph image of an adipose cells, which basically is showing you globules of triacyl glycerols, in yellow, that is occupying most of the volume of such cell. Yeah, so it's packed very, very efficiently. And it turns out that, if you recall, I have been telling that brain utilizes only certain types of fuel. It prefers sugars and ketone bodies when sugars are not available. So fatty acids don't serve as fuel for the brain, okay? Brain will utilize acetyl coenzyme A for doing citric acid cycle, but that comes from somewhere else. So fatty acids cannot serve as fuel for the brain, and that's mainly because fatty acids are typically bound to a protein called albumin in your blood plasma. And when they are bound to albumin, they will not be able to cross the blood-brain barrier. Okay, so something to just keep in mind. So here, I always say that, look, it's not always bad to have some extra weight, especially if you are going on a mission where there'll be scarcity of food. So if you look as the stored metabolic fuel in a 70 kilogram average person, you are seeing the energy per gram of dry weight in different classes, right? From fat to protein to sugar, polymer, etc. And to what is their net amount of weight in your body, right? And how much energy, therefore, they can provide when needed. Okay, so fat is, you can see, 37 kilojoule per gram of energy it can produce. More than anything you can see listed here. And it is in copious amount in your body. It's almost 15 kilogram of dry weight of fat you have stored in your adipose tissues. And energy wise, that's where you have the maximum amount of energy available. More than 500,000 kilojoule energy is available from your fat cells. Proteins are the next class, especially from the muscles. You can see they deliver about 17 kilojoule per gram of dry weight. And they are like almost less than half, about 6 kilogram in dry weight. And you can build proteins, in the muscle if you're, going to do some exercise, etc. So cells can differentiate to produce more muscle cells. But you'll see that proteins also amount for the next most significant part of available energy for your metabolic needs. Okay, look at sugars. Very little. Sugars and sugar polymers, right? Very little. So this is something that you can actually think about it. Now, because my students, they are, you guys are studying biochemistry, and you are learning about metabolism, you often have curiosity to ask, when we eat carbohydrates, why do we put on weight? Right? It's not fat. We are just eating, bread. And it turns out that liver has the capacity to convert excess dietary carbohydrates to fat. Okay, and it can export it to other tissues if the tissues need it for energetic need, or perhaps if it's not needed, it will go and store in the adipose tissues. Okay, muscles do have some amount of fat storage, although we don't talk about it so much, but they do contain some amount of fat storage. Okay, so this is for one reason. You can see why, and I think I will show you how you can actually correlate that when you eat dietary carbohydrates, how you can convert them into fat. And if you think about it, it will all make sense. Okay, if you have excess carbohydrates, eventually you will make acetyl coenzyme A, right? And that acetyl coenzyme A, from pyruvate to acetyl coenzyme A from mitochondrial matrix, it can be transported out of mitochondria to the cytosol by a special shuttle mechanism that we are not going to talk about. Okay, so let me say one important thing. It turns out that fatty acid biosynthesis, which we are not going to do in this class, which will happen in Biochem 2, fatty acid biosynthesis takes place in the cytosol of adipose and liver cells. Okay, so just remember, biosynthesis is happening in the cytosol of adipose and liver cells. Okay, so if you are able to generate acetyl coenzyme A in the mitochondria, it basically is transported to the cytosol via a shuttle mechanism, that involves a molecule like citrate, for example. And once the acetyl coenzyme A comes to the cytosol, then the fatty acid biosynthesis begins. Okay. But fatty acid degradation, catabolism of fatty acid happens in the mitochondria. So you can see how, again, the two processes are nicely regulated, and they can easily be regulated because they are compartmentalized, right? Biosynthesis of fatty acids is happening in the cytosol, whereas fatty acid oxidation, degradation is happening in the mitochondria. So you can imagine that they must be reciprocally regulated. You don't want to carry out fatty acid synthesis when you are actually doing fatty acid breakdown. So this is the beauty of metabolism, how it integrates well, right? All right, so with that little introduction, let's talk about the overall process, how fatty acid, whatever you ingest, right, is catabolized, and how is its inherent energy captured. Okay, so this seems like a busy slide for you, and but we will go over this. You can see that the process has been divided into 8 steps, and we will go over them one by one. Okay. And so one thing, I will tell you that once you ingest fat in your diet, the fat moves through, right, from the stomach to a small intestine. And you will see this very interesting organ called gallbladder. Remember gallbladder? Yeah, gallbladder is a very, very important organ, okay, that stores bile acids. What are bile acids? I think I talked about that, if you remember. Bile acid, an example I'm showing you here on the left hand side, you can see that it has A, B, C, D cholesterol ring, right? But you will see that the D ring has a relatively polar head group, as represented by R2 here, short and small, nice polar group. Okay, so as a result of this you can see that this molecule is an amphipathic molecule, where you have a charged on the R2 group and then a non-polar end. So it almost acts like a detergent. Okay, it's like a detergent, it is. So where are the bile acids produced? It turns out that bile acids are made in the liver, okay, and then they are transported to gallbladder for storage. Okay, and so gallbladder will release bile acids so as to emulsify your neutral fat. What is neutral fat again? To reiterate what neutral fat is, I'm showing you the structure here. It's nothing but triacyl glycerol, long chain fatty acids, ester of glycerol, right? So this molecule is fairly hydrophobic. And so you need something so solubilize it. And that's what bile acid does. Okay. So in the step 1, bile salts will emulsify the dietary fats in the small intestine, and you'll form this mixed micelles like structure. Once you have formed this mixed micelle like structure, it will have access, it will be accessible to lipases. Remember, lipases are the enzymes that are produced in the pancreas, mouth, and stomach, and there are different classes of lipases, and they basically will degrade triacylglycerols into glycerols and fatty acids. Okay, so that's what emulsification does. And once that happens, intestinal lipases have access to triacylglycerols. You will basically break triacylglycerol into fatty acids and glycerol. Okay. And both fatty acids and glycerols are utilized. Okay. Fatty acids, it turns out that you look here, it's very cool how these intestinal epithelial cells are organized and how beautiful they are. It turns out that this intestinal mucosa, which is nothing but the epithelial cells of the intestine, it is very, very specialized. Okay. It cannot assimilate, or it cannot absorb neutral fat all by itself. Okay. It can only absorb fatty acids. Okay. So broken down fatty acids by the intestinal lipases are now absorbed by the intestinal mucosa. Okay. And then once they are absorbed, it turns out that they are reassembled into this species that you are seeing here in the blue called chylomicron. Wow, it's beautiful. It's like a football, but on the surface, you will see certain proteins and certain charged group. And what this is, I will show you the structure of chylomicron in details in a little bit. But basically, this intestinal mucosa has a capacity to take all the hydrolyzed fatty acids and glycerol and assemble that whole thing with the help of certain proteins like ApoC2, here shown here in green. It will assemble the whole molecules of proteins and fatty acids and glycerol into what we call a chylomicron. Okay, chylomicron is nothing but it's an aggregate, right, of fat and protein. So a lipoprotein aggregate. So so that's what you have. That's the step 4. So triacylglycerols are incorporated with cholesterol and certain apolipoproteins. When you say apolipoproteins, means those lipoproteins are not bound to their cognate ligand. That's what apo is added for that. Okay. Anyway, so you can see that chylomicrons. So chylomicrons, then what happens is they move through the lymphatic system and blood vessels. Okay. So they roll along in your lymphatic systems. You know what lymphatic system is, right? Lymph system is a network of these organs that constitute, for example, lymph nodes, lymph ducts, and lymph vessels, right? That make a very important part of your immune system, body's immune system. And so nothing else I can say more than that, other than just to say that lymph is, is a clear to white fluid, basically, where white blood cells are floating around, lymphocytes. Okay, and whenever they see certain foreign viruses or bacteria, they actually attack and kill them. Okay, so lymph system is very, very important system that actually moves immune fluid from tissues to the blood and blood to the tissues. Anyway, so basically they move through, right? And it turns out that when they are moving through the blood vessels, their ultimate target could be muscle cells, which actually need those fats for producing energy, ATP, so it can actually, do muscle contraction, etc. Or if not needed, they can go to fat cells, adipocytes, where they can be stored. Okay. it turns out, think about coolness, right? Wherever you have fat cells or muscle cells, the blood vessels that are lining them, they actually have a specific lipase called lipoprotein lipases that are activated by interactions with ApoC2, okay, the protein that is shown here in the green. Okay, so those lipases that are lining the myocytes and adipocytes, they get activated when they see this protein ApoC2 on the chylomicron surface. And basically they break open the chylomicron. And then fatty acids can then enter those cells. And if it's muscle cells that need ATP, they will get metabolized by beta oxidation. Or if they need to be stored away in adipocytes, they get stored away. Now, chylomicron is, is very interesting actually. And it turns out that if you went to, McDonald's and ordered a triple Whopper, the fattest burger that you can order for additional 10 cents. What will happen once you eat that, fat-rich burger, and if somebody actually looked at your blood literally will be white. It will be milky. You know why your blood will be so milky? Because it will be full of chylomicrons. Chylo means milky, okay? So you can literally see that, but chylomicrons, have a very limited half-life. In almost 8 minutes to 10 minutes, they get degraded, okay? Because the fat must be either stored away or must be metabolized by myocytes, etc. Fun, right? Really fun slide. Yeah. So, so that's the process, okay? And when we talk about amino acid metabolism, I will go through a similar slide. So a little introduction to lipases, although we did talk about lipases, as I said, there are different types of lipases, pancreatic lipases, intestinal lipases. And all they do is they basically take triacyl glycerols and they can cleave them into, they can cleave acyl group either at the C position, a C1 position, or at the C3 position. Okay, so if you cleave both 1 and 3, you can see that you will generate monoacyl glycerol. Monoacyl glycerol can be assimilated by intestinal mucosa for making the chylomicrons. And then you have diacyl glycerols shown here if you cleave either 1 or 3 here. And the other ones can also get degraded by some other lipases. But anyway, so that's what happens. What the structure of chylomicron itself looks like is shown here on the left hand side. So if you look at it, it actually is almost 100 to 500 nanometer in diameter. Okay, so 1/10 of a micron to half a micron, right? And you see here that the surface of chylomicron has phospholipids, right? It's basically not the surface, but the surface is the head group of the phospholipids. So you can see they're showing you all these blue things. They basically head group of the phospholipids, right? And then inside it, you will see that that's where the triacyl glycerol is all sequestered. Okay, so there is a membrane-like structure on the surface, right, which is stabilized by cholesterol. You remember cholesterol actually stabilizes membranes, right? And then you have certain specialized proteins like shown here, B48, C3, and C2. C2 is the one we talked about in the previous slide. Those proteins actually protrude on the surface of the chylomicron, and they are very, very important in recognizing those lipoprotein lipases that line the surface, intestinal surface, inner surface of either a myocyte or adipocyte. Okay. And so you can see that 80% of the mass is inside right there, packed. So it is quite rich in fat. Cool, right? So with that, I want to talk about a little signaling. How, when hormones like glucagon or epinephrine, when they are released, right, into the bloodstream, how they can mobilize triacylglycerols. So stored fat can be mobilized to generate energy to tissues that actually need energy, for example, muscle cells. So if you look at this figure, you will see that on the left hand side is adipocyte, right? On the right hand side is a myocyte, right? Muscle cells. That's where the fatty acid has to go and do beta oxidation, citric acid cycle to make ATP, etc. So how is triacylglycerol mobilized, right? So, whenever there is a low levels of glucose in the blood, you know that it will trigger the release of glucagon, right? And so glucagon will go and bind to the receptor, right, in the, let's say, adipocyte membrane. And once glucagon binds to the receptor, it will, that receptor is a G protein coupled receptor that will get activated. And one of the downstream event will be this enzyme adenylyl cyclase that makes the second messenger, cyclic AMP from ATP, that gets activated. Okay. So binding of hormone to the receptor will stimulate adenylyl cyclase via G protein to make cyclic AMP. And once you have cyclic AMP concentration rising, you know this signal, right? pKa will get activated, protein kinase A, cyclic AMP dependent protein kinase, right? pKa. So once pKa gets activated, it's a kinase, it does two things, okay? One thing it will do is it will phosphorylate hormone sensitive lipase enzymes, okay? It turns out that hormone sensitive lipase enzyme, that's why it's called hormone sensitive, okay? It's only get activated when hormone binds to the receptor. Hormone sensitive lipase is only active when it is in the phosphorylated form. Otherwise, it exists in the non-phosphorylated inactive form. So when pKa gets activated, it will actually go and phosphorylate hormone sensitive lipase and phosphorylate it. And once phosphorylated, that's one thing pKa will do. The other thing that pKa molecule do, enzyme will do, is it will go and phosphorylate a protein called perilipin. Okay, what is perilipin? Well, it turns out in the yellow, you are seeing these lipid droplets, right? Which is nothing but triacylglycerol. It turns out that that triacylglycerol lipid droplet in adipocyte, it is protected by multiple copies of a protein called perilipin. Okay, that's what you are seeing here in purple. It's nicely, it forms a barrier. Okay. So it is nicely protected, but when pK gets activated, pK will go and phosphorylate the protein perilipin, each one of them. Okay. Think about what will happen. Once perilipins are phosphorylated, there will be lots of negative charge surrounding them, and they will basically hate each other. Okay. And they will create porous environment. Makes sense, right? Electrostatic repulsion will actually loosen them up. And so this opportunistic enzyme, hormone sensitive lipase, which is now phosphorylated, can actually access the whole reservoir of lipid droplet and catalyze the hydrolysis of fatty acids from triacylglycerol. Okay, so then in the step 5, you are seeing that now you have individual fatty acids in the adipocytes. So these individual fatty acids simply leave the adipocytes by diffusion. Okay, concentration goes up, they are diffused out, and they go into the bloodstream, right here shown in the orange in the center. And once fatty acids go into the bloodstream, they actually bind the protein albumin in the blood. Fatty acids will not be happy. Remember, they are very hydrophobic. So there are carrier proteins called albumin. Albumin will bind fatty acids, and they will try to take those fatty acids to the target tissues. Could it be myocytes here? Yes. And so there would be, in myocytes, there will be specific fatty acid transporter that will actually try to bring fatty acid in. Once fatty acid is in the myocytes, what happens is that fatty acid gets activated to using coenzyme A, okay, in the cytosol, and then it is brought into the mitochondria for beta oxidation and citric acid cycle and respiratory chain. A lot of good information here that you already know, but if you stare at the slides, everything will be so clear to you, okay? And so that's how you produce, my muscle cells will be able to produce much needed ATP, right? All right, so that's that. Now, what happens to glycerol? Yeah, it turns out that glycerol also has some energy. Okay, 95% of the biological energy of triglyceride is in the fatty acid, but 5% of the energy is there in the glycerol. So what happens to it? It turns out that nature doesn't waste it. There are enzymes that actually will channel this glycerol molecule through glycolytic pathways. Okay, so what happens? You have glycerol here molecules, you can see, right? You can see a secondary alcohol here. And glycerol kinase will basically take glycerol and ATP and make glycerol 3-phosphate. Okay, so it's not glyceraldehyde 3-phosphate because that would be done, right? No, you first make glycerol 3-phosphate from glycerol using glycerol kinase. Then glycerol 3-phosphate gets oxidized to a ketone. Okay, so glycerol 3-phosphate dehydrogenase. Very easy to memorize that and name of this enzyme. Glycerol 3-phosphate dehydrogenase will utilize NAD plus and oxidize that secondary alcohol to a ketone. So now you have a molecule that you can actually recognize called dihydroxyacetone phosphate. The dihydroxyacetone phosphate, if you recall, gets converted to glyceraldehyde 3-phosphate with the help of enzyme triose phosphate isomerase, right? And so the same enzyme will actually convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. And then once you have glyceraldehyde 3-phosphate, then you can go do your payoff phase of glycolysis, right? So you can make ATP. All right, so that's cool. We know where molecules are. So question is, fine. We brought the fatty acid into muscle cells, myocyte. So fatty acids will be first brought to the cytosol, right? What happens once they are in the cytosol? It turns out that they are broken down, but there are steps, okay? Knoop was the first one to show that when fatty acids are metabolized, when they are degraded in a sequential step by removal of 2 carbon unit. And that removal happens in the mitochondria is what was shown by Leninger, the textbook, textbook we follow. So yeah, Leninger is the first one to show that this thing is happening in the mitochondria, the removal of 2 carbon unit. And then 2 other scientists, Leinen and Reichardt, they showed that the 2 carbon that is released from the fatty acid is actually not just the free acetate, but it's an activated acetate, which is acetyl coenzyme A. Okay. And so the way process happens is that you do beta oxidation. And I will show you why the name beta oxidation happens in the next few slides. Okay. So this process literally begins with the oxidation of a carbon that is beta to the carboxy carbon. That's why the term beta oxidation, right? Okay, so remember what I'm telling you. Fatty acids, once they are in the cytosol, right? In the cytosol, they must be activated before they can be carried into the mitochondria where the beta oxidation happens. That's something that you have to keep in mind. But let's look at overall how fatty acids are broken down. So imagine that you have a fatty acid that is already been activated with coenzyme A. That's what you're seeing here on the top. So what you do is you use two coenzymes, FAD and NAD+, and using those, what you do is you generate this species that is shown here in the middle. Okay, and if you look at it, stare at it carefully, you'll see that a new carbon, the beta carbon, is now oxidized to a carbonyl. See here, beta carbon on the top is CH2. They're showing you alpha, beta, gamma carbon. Again, you have to write this to know, otherwise you'll get easily confused. You must write this sequence of reactions if you really want to understand them. Anyway, so that's what beta oxidation is. And once you have created this beta keto group, then you basically cleave the bond between alpha carbon and beta carbon. Okay, so you can see the cleavage, and in that process you generate fatty acids that is activated. Okay, so you can see coenzyme A. So that's why the substrate here in the last step here shown is coenzyme A. So you already have activated fatty acid that is now 2 carbons short and acetyl coenzyme A. And you do this process again and again until you get everything converted into acetyl coenzyme A. So repeated cycle of oxidation at the beta carbon and cleavage of the C alpha beta bond to yield acetate unit, activated acetate unit, and then 2 carbon short fatty acid that is also activated. Cool? Okay. Cool so far. We understand everything now. At least so far what we know. How coenzyme A activates fatty acids for oxidation in the cytosol? Obviously, there has to be an enzyme involved. And so that enzyme is acyl-CoA synthetase. Synthetase. Looks like it will use ATP. Great. So acyl-CoA synthetase will take fatty acid and coenzyme A. With the help of ATP, it will actually activate fatty acid. So how does that process happens? Let's look at it. So what is the idea? In the cytosol, you have free fatty acids. So R is a long chain of aliphatic carbons, right? It's just represented here in the pink to show fatty acid. Now you are also seeing ATP, right? If you see here, the carboxylate ion is attacking the alpha phosphorus of the ATP, right? Alpha phosphoryl group. Remember alpha, beta, and gamma? And so the carboxylate ion is basically attacking that phosphorus, and the leaving group is pyrophosphate. So if you look in the first step, which is catalyzed by fatty acyl-CoA synthetase, you form pyrophosphate and fatty acyl adenylate, right? Fatty acyl adenylate. That fatty acyl adenylate remains bound to the enzyme. Once that happens, coenzyme A sneaks into the active site and position itself next to the carbonyl carbon shown here in the pink and displaces AMP. So you can see AMP is the leaving group here in the second step. And you have formed a fatty acyl coenzyme A and a pyrophosphate. Pyrophosphate, hydrolysis of pyrophosphate to 2-inorganic phosphate, 2-PI, is thermodynamically favorable process. That's why delta G is shown here for that step, standard minus 19 kilojoule per mole. And there is an enzyme that catalyzes the hydrolysis of pyrophosphate. It's called inorganic pyrophosphatase. And so 19 kilojoule will come from the hydrolysis of pyrophosphate. That's one major driving force for this reaction. And so for step 1 and 2, delta G standard is about minus 15 kilojoule per mole. So that's how fatty acid gets activated by acyl-CoA synthetase, and that process happens in the cytosol. Now here is something cool also. It turns out that there are various lengths of fatty acids in our metabolic pathways. Okay. And one thing that want to know that fatty acids that are that have carbon number of carbons 14 or more, which basically constitute majority of our fat that we ingest from our diet, or majority of the fat that is released from the adipose tissues. Those are majority of them are 14 carbon or higher. Okay. And so those fatty acids cannot be directly transported into mitochondrial matrix. There is no transporter that can do that. Okay. Or they cannot diffuse by themselves. On the other hand, if you have short chain fatty acids, 12 carbon or smaller, then they can be carried into the mitochondrial matrix simply by diffusion. Okay, look at the nature, how beautifully this thing is organized. All right, so majority of the fat that we ingest and majority of the storage fat, if it has to be metabolized into the mitochondrial matrix, it just simply cannot be activated by coenzyme A and then brought into the mitochondrial matrix. It turns out that those long chain fatty acids, even when they are activated, they are first converted to a molecule called acylcarnitine. And that acylcarnitine is how they are transported into the mitochondrial matrix. Okay. So, what is the carnitine molecule look like? Carnitine molecule structure is shown here at the bottom. I just show it for the fun of it. Okay. We, many of eukaryotic cells can actually synthesize carnitine, from, let's say, lysine, etc. We can synthesize carnitine as well. Okay. But but carnitine is a very interesting molecule. You need this molecule for fatty acid oxidation. So if you want to burn fat, you need carnitine. Why? Because if your fat is 14 carbon or higher in length, then you will need carnitine to bring that fat molecule into the mitochondrial matrix for beta oxidation. So you need that. So, but it turns out that this molecule is also found in large amount in red meat. Okay, so beef, for example, pork. And so carnitine is found in good amount in those meat products. And it turns out that if you eat too much of carnitine, your liver, unfortunately, liver, is the detoxifying center, right? It has enzymes like cytochrome P450 that metabolizes even carcinogens and try to excrete it into the blood, etc., through the urine, right? So liver, unfortunately, takes carnitine molecule and it basically turns them into this trimethylamine oxide, okay, TMAO. This molecule is bad for you. It actually clogs your arteries. And so you will have heart attack if you eat too much of carnitine. So now you can see a link, for example, between eating too much of red meat, and it can happen not just from carnitine, but of course from fatty acids as well. Remember your LDL has to be low, right? Otherwise it actually forms colonies in the blood vessels and blocks the flow of blood, raises your blood pressure. But carnitine is another molecule, unfortunately, that gets metabolized by the liver to generate this species that actually initiates clogging of the arteries. Okay. So too much of carnitine is no good, right? So I'm not sure, but there was some kind of issues with, eating too much carnitine supplements. People were selling carnitine that if you consume carnitine and if you did eat a lot of fat, you'll be able to burn them, right? You will not be putting on any weight, but it's not as simple. I always tell my children, moderation is the key for things. Don't go extreme either way. Cool, right? This is so important. All right, so let's look how actually then carnitine helps. To see that, I'm showing you a figure here where you are seeing a mitochondrial cell setup. Okay, so you can see that there is mitochondrial matrix on the extreme right hand side, light yellow. Then you have inner mitochondrial membrane. That's where you remember your respiratory chains are actually residing. Yeah, inner mitochondrial membrane, very restrictive, very, very picky what can go through and what cannot. Then you have intermembrane space. That's where proton gradient builds up. You can see that. And then you have outer mitochondrial membrane. And then extreme left-hand side, you have cytosol. Cool? Very cool? Cool, cool, cool. So what happens? I told you that fatty acid must be activated in the cytosol with coenzyme A. And it does happen. So you are seeing on the left-hand side activated fatty acid. Okay. So what happens? It turns out that there is an enzyme called carnitine acyltransferase 1. It's shown here in the blue, that resides in the outer mitochondrial membrane. Okay, so its active site has access to both intermembrane space and to the cytosol. So first reaction that happens by this enzyme is that it will take activated fatty acid in the form of coenzyme A in the cytosol, and then it will take carnitine molecule in the cytosol and do a group transfer reaction. So in the group transfer reaction, what will happen is instead of coenzyme A ester of the fatty acid, you will have now carnitine ester of the fatty acid. You can see carnitine has a hydroxyl group here, right? I'm showing you the chemical structure of carnitine again here. So you can see, oh yes, indeed, it can form an ester. Sure, there is a hydroxyl group right there. So you form that carnitine ester of that fatty acid. Now that acylcarnitine molecule actually can get through the intermembrane space, go through a specific transporter, antiporter more specifically, that will try to bring carnitine, acyl carnitine molecule into the mitochondrial matrix. Okay. Once acyl carnitine molecule is in, the reverse reaction will happen. You see, this is how the cells trick themselves, do the beautiful biochemistry by just doing the reaction in reverse order and getting on with their life. This is how fatty acids are brought in. So carnitine, acyl carnitine will then do another transfer reaction that is catalyzed by carnitine acyl transferase 2, that is there in the matrix. You will regenerate carnitine and you actually regenerate your activated fatty acid by coenzyme A. And that is now ready for beta oxidation in mitochondrial matrix. Carnitine leaves through the same transporter that was there in the inner mitochondrial membrane and goes on and brings another molecule of fatty acid. It keeps doing it until the concentration of fatty acid drops in the cytosol. Now, as I said, fatty acid synthesis happens in the cytosol, so left-hand side. Fatty acid metabolism, which is beta oxidation, happens in the mitochondrial matrix in the right-hand side of this picture. And as I said, once you generate acetyl coenzyme A, it can be brought into the cytosol with a special shuttle system that involves citrate molecule. Maybe we'll talk about that, I think, towards the last class. So when you are doing fatty acid synthesis, you do not want to do fatty acid oxidation. You don't want to bring fatty acid into the mitochondrial matrix. It makes sense. They must be reciprocally regulated. And it turns out that the first committed step in the fatty acid biosynthesis is conversion of acetyl coenzyme A to malonyl coenzyme A, 2-carbon activated ester to a 3-carbon activated ester. But you can see structures are very different here, right? But that's what you have, acetyl coenzyme A, malonyl coenzyme A. That is an irreversible step. That is very tightly, must be very tightly regulated. So it turns out that when you have to not do beta oxidation in the mitochondrial matrix, because you have sufficient amount of ATP, energy status of the cells are okay, but you do have excess acetyl coenzyme A, you basically divert that acetyl coenzyme A into the synthesis of fatty acids and you store that fatty acid. And the first step is conversion of acetyl coenzyme A to malonyl coenzyme A. It turns out that malonyl coenzyme A is a competitive inhibitor of carnitine acyl transferase 1. That makes sense. Why? Because if you are generating malonyl coenzyme A, you don't want carnitine acyl transferase 1 to be active, right? So one of the product of the biosynthesis is actually an inhibitor of carnitine acyl transferase 1. And so beta oxidation pathway bringing of activated fatty acid into the mitochondrial matrix is blocked because carnitine acyl transferase 1 is inhibited by malonyl coenzyme A. Makes sense, hopefully, right? All right. Going a little slow here. We are only at maybe middle of the lecture where we should be doing. But it's fun because we have 2 lectures, right, for this class. So we will be okay. We don't have to rush, any rush. So I am just showing you one more time. You take the fatty acid, right, and you carried out repeated beta oxidation reaction, and you generate acetyl coenzyme A. So if you have a 16-carbon fatty acid, you will generate 8 molecules of acetyl coenzyme A. Those all will be fed into the citric acid cycle. The oxidation of 8 acetyl coenzyme A is a 64 electron event, and you'll generate 16 carbon dioxide molecule. Remember, each acetyl carbon has 2 carbons, so that makes sense. It's not the same carbon that are being released as carbon dioxide, if you recall. But anyway, those 64 electrons are actually embedded in the NADH and FADH2 molecules, and they eventually will transfer all those electrons to respiratory chain that will make ATP. Cool? Oh, there's a question.
I have a question.
Sure.
So in regards to that last slide, so for stage 1, it shows that we have 16 carbons. So doesn't that mean we wouldn't be able to directly perform beta or go through beta oxidation? Like,
it's a repeated cycle of beta oxidation. So each time you run a cycle of beta oxidation, which I'm going to talk about at length in the coming slide, you basically generate 2 carbon acetate that is activated by coenzyme A, and then 2 carbon short fatty acid that is also activated. Remember, I showed you that slide right here.
Right, but
and so you keep doing that again and again.
But didn't the slide after talk about carnitine, right? So didn't that say that it had to be, if it was more than 14 carbons, it wouldn't be able to directly go through beta oxidation? Or maybe I'm just confused.
Yeah, you are a bit confused. That's okay. So fatty acids are fatty acids in the cytosol. Remember, I told you like here, how from adipocytes you can bring a fatty acid into a myocyte on the right hand side. Yes. Yeah. Here it is still fatty acid, but this fatty acid will first come and be in the cytosol of myocyte, right? And in the cytosol of myocyte is where it will first get activated to generate coenzyme A ester. Right. Once it is activated as coenzyme ester, it turns out that if its carbon is more than 14 or more, it basically has to utilize this carnitine transport system, where that, let's say 16 carbon fatty acid in the activated form will first get converted to a carnitine ester, and then reverse reaction will happen in the mitochondrial matrix, and you have regenerated your fatty acid that is activated. This is all the carbons and fatty acids are still there. This will now undergo beta oxidation, and by generating and then generate coenzyme A, acetyl coenzyme A.
So it does go through an additional step if it is over 14 carbons, or is the
that I don't know why the confusion is, because if it is directly if it is 12 carbon or shorter, you will directly make a thioester, and that can sneak in through the mitochondrial membrane. Yes, you're right. If it's 14 carbon or more, then it has to do this carnitine transfer reaction to be able to come inside. So carnitine is just using, carnitine acid transferase 1 and 2. They basically are just trying to bring the fatty acid that is activated, that is more than 14 carbon long, inside by doing this first acyl transferase reaction one time and then doing the reverse reaction. And so now you have generated your fatty acid that is active in the activated form. You are literally just shuttling from outside to inside using these 2 enzymes, carnitine acyl transferase 1 and carnitine acyl transferase 2. Is that a little bit clear?
Makes sense. Thank you so much.
Oh, no, this is not easy. I know it. So it's good you keep it all together. Okay. And I have not talked about beta oxidation fully yet, but I will. It's coming up. I'm just going and showing you again and again. So you see what they're showing you here in stage 1. You basically will be cleaving 2 carbon, 1, and each one of those steps is 1 beta oxidation, a sequence of 4 reactions. Okay. And you will see that you will do that multiple times. If it's 16 carbon, you will do this 7 rounds of beta oxidations to get 8 molecules of acetyl coenzyme A. Okay, so let's look at it. Oh, there is a question here. Which of these compounds is oxidized by a multi-enzyme complex that requires 5 different coenzymes? Oh, we only talked about, I think, 2 enzymes that use that, 5 different coenzymes, right? PDH is one, and the other one was alpha ketoglutarate dehydrogenase, right? So alpha ketoglutarate would be the molecule B, right? If you remember the glutaric acid structure. Yeah. Anyway, let's talk about beta oxidation. It turns out that beta oxidation is a repeated sequence of 4 reactions. Okay. And the strategy in carrying out this sequence of reactions is that you create a carbonyl group on the beta carbon. Okay. And that first three reactions of the 4 will do that. All you are doing is you are creating a carbonyl group at the beta carbon. Remember, I was telling you in this, right? You are taking beta carbon and you are creating a carbonyl. So first 3 reactions do that. And then 4th reaction will cleave that bond between alpha carbon and beta carbon. Okay, so that's the 4th reaction. And the product is you have an acetyl coenzyme A molecule and an activated fatty acid that is 2 carbons shorter. Okay, so those 4 reactions will do that. That's what they will do. So you can see that if there are multiple, methylene carbons in a fatty acids, they will all have to go through this cycle of 4 reactions. And you will see this first reaction, 3 reactions, you have seen them, and you will see them again. They are classic reactions. They will see them again in other pathways. So what is that beta oxidation pathway? Can we look at it individually, the sequence of 4 reactions? So here is your 16 carbon fatty acids, palmitoyl coenzyme A. Okay. So I'm showing you 16 carbon and you will see that chemistry mostly is happening at alpha and beta carbon. You will see that, right? So gamma carbon and beyond, you can just write R group if you want to practice what happens in beta oxidation pathway. If you want to practice by yourself, you can write R. That would be gamma carbon, anything, but definitely keep your alpha carbon and beta carbon, right? Otherwise, you will not be able to enjoy the mechanism. So carbonyl carbon is carbon number 1. And next to carbonyl carbon is the alpha carbon. And next to the alpha carbon is the beta carbon. Okay. So if you take palmitoyl coenzyme A, right, it is in the mitochondrial matrix. The first reaction that will happen on this molecule is catalyzed by acyl-CoA dehydrogenase. Why do we call acyl-CoA? Think about it. It's a fatty acid, right? So you have seen acyl group, RCO, right? And it is activated by coenzyme A. So that's why acyl-CoA dehydrogenase. It turns out acyl-CoA dehydrogenase uses FAD as the coenzyme, not NAD. Don't write NAD plus here. It's not. It doesn't bind to NAD plus. So it will only bind to FAD. So it will take FAD and basically oxidize that carbon, alpha carbon and beta carbon signal bond to a trans double bond. Okay, that's what it will do. So that's why the product is named as trans delta 2 enoyl coenzyme A, right? If there is an ene and oil, the carbonyl, that's why enoyl, and it is at Remember the fatty acid nomenclature? At the second carbon is where the double bond is, and therefore delta 2, superscript 2. I hope you are connecting this, right? We talked about this at length. So that's the molecule you make. And then this molecule becomes a substrate for enoyl-CoA hydratase. As hydratase means it will add water molecule. It will help add water molecule in this molecule, which is trans delta 2 enoyl-CoA, in a stereoselective fashion. And because you will be generating a chiral center at the beta carbon now, because you are adding water. That's where the water adds. So that's why they are telling you the product name is L-beta hydroxy acyl coenzyme A. At the beta position, you have hydroxy group. If L is bothering you, don't worry about it. L is usually what you always have L-mylate. That's why L here. So, but it is basically beta hydroxy acyl coenzyme A. Again, practice it only in writing. That's the only way you will be able to memorize this and enjoy this. In the next step is the third enzyme called beta hydroxy acyl CoA dehydrogenase that will utilize NAD plus as the coenzyme and make that secondary hydroxyl group to a ketone group, oxidize it. Oh, so you got beta keto acyl coenzyme A. And in the fourth reaction is the beta cleavage of the beta keto ester. It's a reverse of the Claisen condensation reaction, if you recall. And so this is the cleavage of beta keto ester catalyzed by acyl CoA acetyl transferase, also known as thiolase. If your big name is bothering you, you just remember the name thiolase. Thiolase, why? Because it actually utilizes coenzyme A as one of the substrate, and it will cleave that alpha carbon beta carbon bond, and beta carbon will be then activated with coenzyme A. So that's what's 14 carbon acyl coenzyme A. That will be myristoyl coenzyme A, because you started with palmitoyl coenzyme A. So 14 carbon activated fatty acid and 2 carbon activated acid, which is acetyl coenzyme A. This 14 carbon activated fatty acid, myristoyl coenzyme A, becomes the substrate, goes back and does the same thing again and again until you have all the molecules converted to acetyl coenzyme A. So if you took the 16 carbon fatty acids, saturated fatty acids as shown here, you will have to do 7 rounds of beta oxidation. One is shown here on the left hand side, and then the remaining are shown here in the right hand side. So 1, 2, 3, 4, 5, 6, 7. Last step, you'll generate 2 molecules of acetyl coenzyme A, right? When you have C4. So that's why 7 rounds. That's your beta oxidation pathway right there. Cool. And so we'll discuss the individual reaction real quick, right? So this is, you can see a fatty acid. First thing you need to do is you have to actually take, oxidize that single bond to a double bond, right? And so you must move hydride, right? You must remove electron, right? So it turns out that enzyme has utilizes a general base. I believe it's a glutamate carboxylate group from the side chain of acyl CoA dehydrogenase. It will pick up that proton, and then you can see the arrows are being pushed. The way arrows are being pushed here, you know that that hydrogen at the beta carbon is leaving as a hydride with its electron pair, right? And that's why you generate FADH2, and you actually have generated a trans double bond. Cool. So that's first step. The second step is catalyzed, right, by enoyl-CoA hydratase, also known as crotonase. And that is basically, it's just the addition of double bond in a stereoselective manner. And you have seen that reaction, many times, if you recall. Fumarate, for example, adds to stereoselectively water molecule to generate mylate and all that, remember? So yeah, so then you produce L-beta hydroxyacyl coenzyme A. In the third step, you basically oxidize that secondary alcohol using NAD plus, and that reaction is shown here. You basically generate, again, it will be a hydride transfer, right? How will you push electrons here if you were to show? I'm not sure if I am showing you the mechanism of these, but basically what you will do is you'll pick up that proton and move that, right, to generate carbonyl, and then you remove that hydrogen at the beta carbon as hydride, right? So you generate carbonyl. I'm just saying that you could do that, right, in terms of just formally arrow pushing. And so in that process, you generate NADH. Remember, each NADH molecule can go and transfer electrons and make 2.5 ATP molecule. Remember? So that's something that you should always see when you produce NADH. Fourth reaction is the thiolase reaction, where I will show you what happens rather than just discussing that. So this enzyme thiolase has a cysteine residue in the active site. It will take beta keto acyl coenzyme A, right? And you can see, imagine that it can attack just like chymotrypsin mechanism. Chymotrypsin, you are using serine hydroxyl group. Here you are using cysteine residue, sulfhydryl group. You will generate this tetrahedral intermediate that can collapse, right, to generate this acetyl coenzyme A, yellow molecule that you need, right? But at this point, enzyme is still tied up in the form of thioester. They're just showing you a few different steps. And you can see why they are showing you a few different steps. You will be able to form that carbon ion because you can resonance stabilize that carbon ion into the carbonyl oxygen, right? That's why that reaction happens. See, the chemistry is so fascinating, right? That's what I always tell my students. Yeah, it's biochemistry, but look at the beauty of enzymes, how they're carrying out the reactions. In any case, you have formed, just like how you form acyl enzyme intermediate with the serine hydroxyl group of the catalytic nucleophile in chymotrypsin, you are basically have formed a thioacyl enzyme intermediate in this case, which then gets attacked by coenzyme A, and enzyme leaves, and you have generated a 2 carbon less activated coenzyme A ester of the fatty acid in the fourth reaction. All right, so what is the summary here? Repetition of the cycle yields a succession of acetate units. See how loosely they're using this word, or I am using this word acetate unit here. Actually, it's an activated acetate, right? So just keep that in mind. Sometimes we do have problems of using things informally a little bit. So if you have 16 carbon fatty acid like palmitic acid, then you will get 8 acetyl coenzyme A. It turns out that if you actually did some accounting, you will see that complete beta oxidation of one molecule of fatty acid, here in this case 16 carbon fatty acid, saturated fatty acid, that will yield about 108 molecules of ATP. If you recall, when we talked such stoichiometry, when we were talking about glycolysis, one molecule of glucose actually yields close to about 30 to 32 ATP molecule, right? You can see here, 16 carbon fatty acid, one molecule yields like, more than 3 times, 108 molecules of ATP. And that's because this molecule of fatty acid is in the most reduced states of the carbon, right? So there are lots of electrons embedded in these molecules, very energy rich. And so that's what makes this, fuel of choice for migratory birds and many other animals, such as seals, such as polar bears. So yield of ATP during the oxidation of one molecule of 16 carbon activated fatty acid is shown here. What are they showing you here? If you look at this, they are showing you the enzymes that are involved in the oxidation of fatty acid, all the enzymes, all the way going through the citric acid cycle. So you can notice here that you have acyl-CoA dehydrogenase, right? That utilizes FAD. So you will make 7 molecules of FADH2. Think about it. Very cool. This is 16 carbon fatty acid, right? But you need to run only 7 rounds of beta oxidation cycle, right? So that's why 7 FDH2 molecule. And each FADH2 molecule makes 1.5 number of molecules of ATP. That's why you are making 10.5 ATP. The second enzyme of the beta oxidation, beta hydroxyacyl-CoA dehydrogenase, that utilizes NAD, will make 7 molecules of NADH. And each molecules of NADH makes 2.5 molecules of ATP. So that's why that number. Then once you have generated all 8 molecules of acetyl coenzyme A, then you will take that 8 molecules of acetyl coenzyme A through citric acid cycle enzymes. And so isocitrate dehydrogenase is utilizing 8 molecules of NADH, not 7. That's why 8 here. And then that's why you'll get 20. And you sum them up, all these enzymes of citric acid cycle enzymes, you will see that you are able to generate 108 molecules of ATP with 1 molecule of palmitoyl coenzyme A. This is the fatty acid that is already activated. Okay, so
Professor,
yes,
just a quick question.
sure.
What if your fatty acid is an odd chain fatty acid and it's not even? You're not going to result in the two acetyl-CoAs, right, at the end?
Very good question. Yeah, so we'll talk about that a little bit later, I think, in the next class.
Okay.
It turns out that most of the metabolism is derived with even chain fatty acids, but there are certain species that will utilize. And yes, when you have odd chain fatty acids, instead of acetyl coenzyme A, you will make propenoyl coenzyme A as the last product, and that gets utilized as well. So yeah, we can't, unfortunately. I mean, there are lots of good nuances to fatty acid metabolism, but excellent question, and thank you for asking. Okay, so one thing that I wanted to, I think I could, close it here just with this thought that remember, when you metabolize fatty acids, you generate carbon dioxide, but you also generate metabolic water molecule, right? Because molecular oxygen will accept electron and be converted to water. So certain animals that actually live in desert, dry climates, they're showing you gerbils here, and killer whales which actually live and dive in deep sea, deep ocean, and camels. Camels, the hump that you see in the camels, it's actually a large deposit of fat there. Okay, killer whales, they don't drink seawater, right? So their only source of metabolic water is the oxidation of fat that they have in their adipose tissues. So metabolic amount of water, can be are produced in many of these animals that use fat as fuel, and this is very useful when drinking water is not available for them, right? So this is something that you want to keep in mind. And so that's where I think I will end today's lecture, and we'll talk about the second part in the next lecture on Tuesday. Let me stop the recording here.